Hi everyone, today we're going to be talking about cell division, specifically focusing on eukaryotic organisms. So we'll do an introduction, we'll talk about chromosomes, and then we'll look at our two types of cell division, mitosis and meiosis. When we talk about meiosis, we'll also look at how meiosis helps generate genetic diversity within populations. We have two basic reasons why cells are going to divide. The first is reproduction, and we'll see this in both our unicellular and multicellular organisms. The second reason why cells need to divide is for growth and repair, and this we'll see in our multicellular organisms where they're growing in size or repairing any damaged cells or tissues. We have two types of cell division, mitosis and meiosis. Mitosis is going to result in two genetically identical daughter cells. They'll be genetically identical to each other as well as to the original parent cell. Meiosis, on the other hand, is a specific type of cell division that is used in sexual reproduction, and it is used to generate our gametes, which is the egg in females and the sperm in males. The egg and the sperm then fuse together to form the very first cell known as the zygote. Let's talk a little bit about reproduction. We have two types of reproduction, asexual and sexual. In asexual reproduction, a single cell or single individual is going to divide to produce its offspring. So the offspring produced are going to be genetically identical to the parent. We see this in bacteria and archaea, but we also see it in some eukaryotes such as sponges and sea stars. Sexual reproduction, on the other hand, requires two individuals, and these individuals are going to be of opposite mating types. They're going to come together, their genetic information is going to be combined in the offspring. And we'll see this type of reproduction in most eukaryotes. So why is sexual reproduction important? Well, it's important because it increases genetic diversity within populations. Think about it like this. If we had a single population and all individuals in that population were carrying the same genetic information, and there was some kind of change in the environment, maybe a drought, maybe a disease comes about, everyone is the same, and let's say nobody's resistant to this change, well, that's going to wipe out the entire population. However, if we have a population that is genetically diverse and we have individuals carrying different genetic information, there's a greater chance that when that change comes about, we'll have individuals that are resistant to that change, are able to survive and able to continue that population. So the example we have up here is that of potato blight. In the 1800s, potato blight caused the Irish potato famine in Ireland because all their crops were genetically identical. They were using cloned potatoes, and so when that water mold infected the potatoes, all their crops were wiped out and it resulted in widespread famine. Had they had genetically diverse crops and potatoes with different DNA, some of those potatoes may have been resistant to the water mold, and they would have survived, and it wouldn't have wiped out all of their food source. All right, let's talk about chromosomes. So we know that genetic information within the cell is stored inside the DNA. Our DNA is double-stranded. So we have two polynucleotide chains connected to one another. Those polynucleotide chains are wound around an imaginary axis, and that forms this helical structure known as the DNA double helix. Now we have lots and lots of DNA. We need to fit that DNA into a very small space inside the cell. So we need to compact it as much as we can. So that DNA double helix is wound around special packaging proteins, condensing it even further. Together, the DNA and those packaging proteins, that is known as chromatin, and that is how the DNA is found within cells that are not actively dividing inside our bodies. Once cells start to divide, that chromatin is going to condense even further, and that's when we form the chromosome. So that's important so that when the DNA is separated during cell division, we don't have any breakaging. So think about this. Think about having a large plate of spaghetti and it's all entangled and you want to separate different strands to different sides of your plate. Well, some of the strands may get tangled, they may break, and so you're going to get breakages in that spaghetti. But if each of those strands of spaghetti was wound up tightly into a little ball, well, it's going to make separating it much easier and, of course, prevent any kind of breakages from occurring. So same thing with the DNA. It gets wound up really, really tight so that it's easy for it to separate during cell division. So before cell division even occurs, our DNA is going to get duplicated. So let's have a look at this diagram over here. Here we have a chromosome. The DNA gets replicated and it's an identical copy of this original strand of DNA. This identical copy of DNA remains attached to that original strand at the centromere. I always think centromere, attach here, so it remains attached. Now this entire thing is known as a chromosome. This strand of DNA is identical to this strand of DNA, and each of these is known as a sister chromatid. Within our somatic cells, which are our normal body cells, such as our skin cells, our eye cells, our hair cells, these cells are going to be diploid, meaning that they contain two sets of chromosomes. One set you would have inherited from your dad, one set you would have inherited from your mom. So we have these pairs of chromosomes, and these are known as homologous pairs of chromosomes. A homologous pair of chromosomes will carry the same genes, and those genes will be at the same location, but we could have different versions of those genes. Those versions are known as alleles. The chromosomes that come from your dad are going to be known as paternal. The ones coming from your mom are going to be known as maternal. So if we look at ourselves, we have in each cell 46 chromosomes, which means that we have 23 pairs of chromosomes. 22 of these chromosomes are known as autosomes, and they are the same length, they have the same genes at the same location. The 23rd pair, however, is our sex chromosomes, and we have an X chromosome and a Y chromosome. The X chromosome is longer than the Y chromosome, so here's our X, here's our Y. The X chromosome is longer than the Y chromosome and carries a couple of different genes compared to the Y chromosome. These sex chromosomes determine the sex of the individual. If the individual is carrying two X chromosomes, they are female. If they are carrying an X and a Y chromosome, then they are male. Now, our gametes that we produce using meiosis are going to be haploid, just having one single set of chromosomes, which means that one chromosome from each of these pairs is going to end up in that gamete. So having a look over here, we have a chromosome and another chromosome. This one is from mom, so it's maternal. This one is from dad, so it's paternal. And let's say that they are carrying the same genes at the same location. That would mean that they are a homologous pair of chromosomes. Before cell division occurs, these will be duplicated, and those duplicated copies will remain attached to that original strand. So if we have a look over here, now we call this whole structure a chromosome, and together these two chromosomes still make up our homologous pair of chromosomes. If we look at this chromosome over here, we have our sister chromatids. Over here, we also have our sister chromatids. If we had to compare this one and this one, those would be non-sister chromatids. Although they're carrying the same genes at the same location, remember they may be different alleles because this one was originally from mom, this one was originally from dad. Alright, so let's talk about mitosis. Mitosis is going to be more complicated than binary fission, which is the cell division that we see in our prokaryotes. And the reason for this is because eukaryotes have a lot more DNA than prokaryotes, and that DNA is located inside the nucleus. So mitosis is going to take one single parent cell and it's going to produce two daughter cells. Those daughter cells are still going to be diploid, and they're going to be genetically identical to one another as well as to that original parent cell. So we're going to see mitosis used in our unicellular eukaryotes such as yeast, and they will be using it for asexual reproduction. We can also see it used in some multicellular organisms for asexual reproduction as well, such as our starfish and our sponges. All multicellular organisms are going to use mitosis to grow in size and to repair any damaged tissues or replace damaged cells. Mitotic cell division as a whole is going to have two stages. The first stage being mitosis, and this is where the nucleus is going to be divided. And then the last stage is going to be cytokinesis, and this is going to be the division of our cytoplasm. Cytokinesis does overlap with the end stage of mitosis. Alright, so we're going to have a look at the stages of mitosis now. So we have 4 stages in mitosis: prophase, metaphase, anaphase, and telophase. Some people like to remember the order of these stages using the acronym PMAT. Let's go through each of these stages. Firstly, prophase. What's going to happen in prophase? Well, our nuclear envelope is going to start to break down. It's going to start to break down. Our chromatin is going to condense and form chromosomes. So let's pretend we're dealing with a cell that only has 2 homologous pairs of chromosomes. So it has 2 chromosomes from mom and 2 chromosomes from dad. So the chromatin condenses and we form our chromosomes. Remember that the chromosomes are in the duplicated state because even before we enter mitosis, the DNA is going to be replicated. So we have our 2 sister chromatids attached to one another at the centromere. The other thing that's going to happen in prophase is that our centrosomes are going to start to move to opposite poles of that cell, or opposite ends of the cell. Centrosomes are part of the cytoskeleton. When we spoke about the cytoskeleton at the beginning of this course, I mentioned that they're going to be involved in cell division. The other thing that's going to happen during prophase is that spindle fibers are going to begin to form. Spindle fibers, once again, are part of that cytoskeleton, and they're going to help our chromosomes move around the cell. All right, in the next stage, metaphase, our nuclear envelope is completely gone, and our chromosomes are going to line up at the center of the cell, and they're going to line up in single file. Our centrosomes are now at opposite poles of the cell, and our spindle fibers are going to attach to the sister chromatids. So spindle fibers from this side of the cell will attach to sister chromatids on this side of the chromosome for each of these chromosomes. And then spindle fibers coming from this side of the cell will attach to the sister chromatid on the other side of the chromosome. So you can see that those spindle fibers are attached to each of those sister chromatids. So when they start to pull apart, they're going to split those sister chromatids apart. And that's what happens in anaphase. So our spindle fibers, they will begin to shorten and this will actually pull those sister chromatids apart and move them to the opposite poles of the cell. Just remember that now that we've pulled those sister chromatids apart, they're once again just known as a chromosome. Then we move into our last stage, which is telophase. So here our chromosomes have arrived at the opposite poles, and our nuclear envelope is going to begin to form at each of these poles around those chromosomes. our spindle fibers are going to also break down, and our chromosomes are going to start relaxing again and forming the chromatin. The other thing that's going to happen in this stage is that cytokinesis is going to start. So we're going to start dividing that cytoplasm. And then obviously the cell will move into cytokinesis and the cell will split into two cells. Each of those cells are genetically identical to each other as well as that identical to that original parent cell. Alright, so now let's talk about meiosis. Meiosis is the cell division that we're going to use in sexual reproduction. It's very important because what it does is it's halving the chromosomal number when we form those gametes, which means that when our gametes fuse back together and form the zygote, we get back to the original chromosomal number. We don't double the chromosomal number each time we reproduce. So meiosis is going to take one single diploid parent cell and it's going to divide, and by the end of it, we'll actually have 4 cells, and each of those cells will be haploid. They will obviously not be identical to the parent cell, but they're also genetically unidentical to one another. Once that egg and sperm fuse together, we form our zygote, which is diploid, and that zygote will then continue to grow and divide using mitosis. So meiosis only used to create the gametes. Meiosis has 2 rounds of division. So we have meiosis 1 and meiosis 2. If we have a look at this cell over here, so we once again have just 2 sets of chromosomes. Here's a homologous pair of chromosomes, which is going to say the blue is from dad and the pink is from mom. And here is another homologous pair of chromosomes. Meiosis 1 is going to split up our homologous pairs of chromosomes. So after meiosis 1, the cells are already haploid. They have half the original chromosomal number. However, if you have a look, those chromosomes are still in the duplicated state. And that is why we need to go through a second round of division. So meiosis 2, this is where we split up sister chromatids. Sister chromatids get split up, and now we have 4 haploid cells, and the DNA is no longer duplicated. So here are the stages of meiosis 1 and meiosis 2. They're very similar to mitosis. So we have prophase 1, metaphase 1, anaphase 1, telophase 1. And then we go into prophase 2, metaphase 2, anaphase 2, and telophase 2. One very important difference I want to point out to you is that during metaphase 1, instead of lining up in single file at the center of the cell, the chromosomes are going to line up in their homologous pairs. So our spindle fibers are actually going to attach to an entire chromosome. And that's why when the spindle fibers shorten, we separate homologous pairs of chromosomes in meiosis 1. Meiosis 2, however, during metaphase, they line up single file again, just like in mitosis. So they line up single file at the equator, and then when those spindle fibers separate, we are separating our sister chromatids in meiosis 2. All right, let's talk a little bit about genetic variation. So genetic variation in a population can be brought about in a number of ways, but the ultimate source of genetic variation is mutations. And a mutation is an error that occurs during DNA replication. Most mutations don't really bring about a change in the organism, or sometimes they're detrimental to the organism. However, when a beneficial mutation arises, that's when evolution is going to pick up on that mutation and push it through a population. So mutations are going to be how different alleles or different versions of genes are brought about. The next way that genetic diversity is introduced into populations is actually through meiosis. And we have 2 things occurring in meiosis that are increasing genetic diversity. One is crossing over, and the other is independent assortment. And we'll talk a little bit more about both of these coming up shortly. Lastly, random fertilization also helps to increase genetic diversity in population. So it's completely random which sperm and which egg end up fusing with one another. It doesn't matter what kind of genes they're carrying. That's not going to determine whether they can actually fuse together. All right, let's look at those two processes in meiosis that increase genetic diversity. So the first one is crossing over, and this is going to occur in prophase 1 of meiosis 1. What is going to happen is our homologous pairs of chromosomes are going to find one another, and they're actually going to exchange pieces of DNA. Obviously, they're exchanging the same gene, all right? So if we have a look at this after crossing over, this was the original paternal homologue, and we can see that this strand over here is now different. This is known as the recombinant DNA. So we have that original strand from dad, but now we have this little piece from mom. And if mom was carrying different alleles, this is going to be different to this original strand of DNA now. And down here, here's our recombinant piece of DNA. So we have that original strand from mom with now this little piece coming from dad. And that's going to create genetic diversity and cause each of these cells to be different from one another. Independent assortment, this is also going to happen in meiosis 1. And what happens here is that when those homologous pairs of chromosomes line up at the equator, it's completely random which side of the equator they end up on. There's no law that says all paternal must be on the left and all maternal must be on the right. So it can be a mix match, which means that when they pulled apart during anaphase 1, it's completely random whether the original paternal or maternal homologue ends up in each cell. So if we think about ourselves as humans with our 23 pairs of chromosomes, with all the number of alignments we can have at that equator, we have more than 8 million possible combinations. So just increasing that diversity. So just having a little overview of mitosis and meiosis in humans. So we're going to use meiosis to generate our gametes. Males will generate sperm, females will generate the egg. Remember that the sperm and the egg are now haploid, having half the chromosomal number. The sperm and the egg will fuse together during fertilization and form the very first cell known as the zygote, and this zygote will now be diploid, so having the two sets of chromosomes once again, one from dad, one from mom. From that point onwards, the zygote will develop using mitosis. So to use mitosis to become the embryo and then the fetus and then to become the full-grown adult. And of course that adult will still be using mitosis to repair and replace any damaged tissues and cells. Alright, so take a minute, pause the video, do these review questions, and then we'll come back and go through the answers together. Firstly, looking at the difference between asexual and sexual reproduction. So in asexual reproduction, we just need one single individual, and all the offspring produced are going to be genetically identical to that original parent, as well as to one another. Whereas in sexual reproduction, we have this combining of genetic information from two individuals, and so that's going to increase genetic diversity. Looking at the next question, contrasting mitosis and meiosis in terms of firstly the number of cells produced. Well, mitosis produces 2 cells and meiosis produces 4 cells. If you struggle to remember that, just think of mitosis has a T in it, T for 2, so mitosis produces 2 cells. Next question, the number of sets of chromosomes in our daughter cells. So mitosis, we still have those 2 sets of chromosomes, whereas meiosis, we have a single set of chromosomes. So cells produced through mitosis are diploid, cells produced through meiosis are haploid. And then what about the genetic diversity of the cells? Well, in mitosis, we're all genetically identical. In meiosis, we're all genetically different. And then lastly, how is genetic diversity increased in a population? Well, ultimately through mutations causing changes in DNA, which results in different alleles. Secondly, during meiosis, where we have crossing over and independent assortment. And then thirdly, due to the fact that fertilization is completely random.